You're watching Coronavirus in Context. I'm Dr. John White, Chief Medical Officer at WebMD. There's a lot of talk about vaccines. Some folks are saying it's going to be available by the fall. Others are saying it's years away. To get insight into how realistic vaccine development is, I've turned to Dr. Paul Offit. He is the Director of the Vaccine Education Center and Professor of Pediatrics at CHOP, Children's Hospital of Philadelphia. Dr. Offit, thanks for joining me.
My pleasure.
So where are we on the possibility of a vaccine? Is 12 to 18 months realistic?
Well, remember, the average length of time it takes to do the research of development on a vaccine is about 20 years.
That's 20 years, okay.
If you go through all the steps, that's about how long it takes. If you're trying to get a vaccine out in 12 to 18 months, then you're going to have to skip some steps, major steps.
Okay. What are those steps that might be skipped?
So normally what you would do is you would do a proof of concept. You would have an animal model that would get sick when you inoculated it with COVID-19. Then you would try one or another strategies and see which one worked. Then what you would do is you would do dose ranging trials, trying to see what dose I can give to induce an immune response that at least in animal models correlated with protection. Then you do larger and larger trials, usually hundreds or thousands of people, to do a big dose ranging study. Then you do a phase 3 trial, the definitive FDA licensure trial.
Sure.
So what would you skip? I think you skip the animal model studies. You do small dose ranging studies, which Moderna, which is making an mRNA vaccine, has already done, or Inovio, which is making a DNA vaccine, is in the midst of doing. And then you say, okay, here's a dose that induces an immune response similar to that occurs after natural infection. I'm going to assume that that immune response is protective, even though I don't necessarily know that.
Right.
Then what you would do, a prospective placebo-controlled trial of 1,000 or 3,000 or 6,000 people, which is may or may not be enough to tell you whether it's effective. It's certainly not enough to tell you whether or not you have any sort of uncommon side effect problem. You're willing to accept that when that vaccine is being given, you haven't done a large safety and efficacy trial. So to some level, it may not be as effective as you think, or may not be as safe as you think, but you're scared of the virus. I mean, you're still panicked about the virus because it's still killing 1,000 or 2,000 people every day. Then you're willing to accept some level of risk. If at the time the vaccine is introduced, it's killing, say, 10 people a day, you may not be willing to accept that risk. And so I think it's all risk-benefit in medicine, right? I mean, we give cancer chemotherapies that can kill people to people who have cancer because you're willing to take that risk because the cancer could kill you.
But we're often not having that narrative in the media. We're just talking about a vaccine being available. And you're right, there will be caveats to that in a discussion of risk versus benefit. And sometimes people misestimate the risks of vaccines.
Right. So I think you at some level have to manage expectations. I think that if you haven't given this vaccine yet to tens of thousands of people, so you can't say with confidence that how effective it is, or you can't say with confidence that it doesn't have an uncommon side effect, then you have to manage that expectation.
Would we give it to children, or do we need to study them separately?
I certainly wouldn't give it to children until you are very, very confident that this vaccine is safe and effective, because children don't die from this virus. So you better make sure that you have a safe product before you put it into children.
Hey everyone, it's Dr. Z. So I don't know if you guys have heard about this coronavirus thing. Apparently it's in the news. But my guest today, he's a world expert in this stuff. He is part of a working group working on a coronavirus vaccine. He's the dean of the School of Tropical Medicine at Baylor. Welcome back to the show. You should see our previous interview. This guy is awesome. He's a personal hero of mine, Dr. Peter Hotez. So let's move on to vaccine, because I think people are really interested in hearing from you, the expert on this, who's working on this. What are the prospects for a vaccine? What's the process? What's the time frame? And how effective will it be? And was SARS a good primer for this, given the work done on a SARS vaccine that never ultimately was fully implemented because SARS mostly was controlled?
Well, we learned a lot about after SARS in 2003 and then MERS in 2012, we learned a lot about what we need to do to make an effective and safe coronavirus vaccines. We developed a prototype vaccine based on SARS that was both highly effective in laboratory animals at preventing challenge infections and was also very safe. And I'll come back to that safety issue in a minute because it's a huge one. The other piece to this is there's a particular problem with coronavirus and respiratory virus vaccines. It turns out that certain respiratory virus vaccines can trigger, through mechanisms that we don't entirely understand, something called immune enhancement, where you get eosinophils filtering into the lung, and it actually will make things worse. And what we found was that when scientists started making the first generation coronavirus vaccines after SARS, using killed vaccines or even the whole spike protein from the virus, it actually made things worse. The relevance here is the regulators, the FDA, the scientists at the FDA are going to want to be very careful and very cautious how they proceed with clinical trials, because what happens when you do a phase 1 trial or a phase 2 trial in healthy volunteers in an area where you have sustained community transmission? Will some of those vaccinated individuals, will they develop immune enhancement? And so that's going to be looked at very closely, and it's really going to slow things down for vaccine development. So unfortunately, I think that's a long way to say we're not going to have a vaccine in time for this epidemic.
See, that's really important. And one thing I wanted to ask you about that was, when you say immune enhancement, does that mean you get that phenomenon after vaccination without infection, or when you get infected?
No. What happens is you get vaccinated, and then you're exposed to the virus,
right?
Being in the community. And in the case of RSV, that first generation RSV vaccine, that's what happens. The kids did actually worse than the non-vaccinated kids. So they had to go back to the drawing board. It actually killed vaccine development for RSV for a generation. Now the Gates Foundation and others are trying to support new generation RSV vaccines.
Yeah, and we have not seen that with influenza vaccination, though.
No, it doesn't seem to be a problem with influenza vaccines.
Yeah.
Or at least the ones we use for seasonal flu, but has been a problem with RSV, and in laboratory animals, it's been a problem for coronavirus vaccine. And in laboratory animals, it's been a problem for coronavirus vaccines.
Hey everybody, Dr. Zubin Nemanja. I have a super special guest today in the era of COVID hysteria. This is one of the leading experts on vaccine science, infectious disease, and general rational thinking, Dr. Paul Offit. Let's cut to what are the pitfalls? Now, I had Peter Hotez on the show talking about his work on SARS-2 coronavirus vaccine. Talk about the vaccine. Just hit me with your understanding and where you think this is going, and let's go from there.
I think that if you're going to start testing this vaccine, you have to damn well make sure it's safe. Because remember, the people that are now getting this vaccine in Seattle are unlikely to die from this virus. I mean, they're young, healthy people. So you have to, I just worry that you would in any sense rush this, that you wouldn't do extensive animal model testing. I just worry that you would in any sense rush this, that you wouldn't do extensive animal model testing. I just worry that you would in any sense rush this, that you wouldn't do extensive animal model testing, that you wouldn't then do extensive phase 1 safety and immunogenicity testing before you ever get to an efficacy test. I worry that people will line up to try and get a product that I think has not yet reached a level of testing that it should reach, remembering that the people who are going to be getting this vaccine are very unlikely to suffer seriously from this virus. So you better make sure it's safe before you put it into them.
Now, one piece of that Peter Hotez brought up was this idea of immune enhancement with respiratory viral vaccines like RSV. What's your thinking? Can you describe that to us and tell me what you think about that with this coronavirus?
Right. So in the 1960s, there was an attempt to make a respiratory syncytial virus vaccine. That's a virus that infects everybody. It's a respiratory virus, but it primarily kills the young, especially prematures, but primarily kills children in the first couple of years of life. And we'll have a few thousand deaths every year from this virus. The way that they made that vaccine, and it was out of NIH in Bob Chanick's lab, was they took the virus, killed it, and then inoculated people with a whole killed virus. What they found was that when then children were naturally exposed to the circulating virus, the natural virus, so-called wild-type virus, they actually did worse if they got the vaccine than if they didn't get the vaccine. That's one example. Another example was there was a whole killed measles virus. It was introduced in this country in 1963. That too, when you were then exposed to the natural virus, actually you had a worsened syndrome, more likely to get pneumonia than if you had never gotten that vaccine. That's a bad sign. And dengue is another example, the so-called Dengvaxia vaccine that's made by Sanofi Pasteur in children who have never been exposed to the virus before. You actually had the so-called facilitated antibody entrance. So in other words, that you develop these sort of binding antibodies so that when you then were infected with when you were infected with actual wild-type dengue, natural dengue, that actually those antibodies facilitated virus entry into cells and made you worse. So there's three examples right there of so-called immune enhancement. And if there's animal, as Peter said, and I'm glad he was willing to come on your show and say that was an issue. Because it's important to know that. I think it's important for the public to know that there may be issues with immune enhancement, which is to say that if you get the vaccine and then you get are exposed to the wild type or natural virus, that there may be a worsened response than if you'd never gotten the vaccine, which is why I think you have to have extensive animal model testing and very slowly move forward on sort of safety testing, safety first. Which is why I think you have to have extensive animal model testing, and which is why I think you have to have extensive animal model testing, and which is why I think you have to have extensive animal model testing and very slowly move forward on sort of safety testing, safety first. I mean, safety, and then immunogenicity testing. But maybe I'm just a bad sport. I mean, I worked on a rotavirus vaccine for 26 years before that became a vaccine, and that's about average for how long it takes to make a vaccine. When Dr. Fauci says, 12 to 18 months, I mean, that is ridiculously optimistic. And now we're already, putting this vaccine into people. I just worry about this a little bit.
Everybody talking about, hey, how are we going to develop this vaccine? Because we'll never get off these lockdowns and these rotating re-outbreaks and all of that, even if we get it under control, if we don't develop a definitive strategy, which in their mind is a vaccine. Walk us through this. What do we expect here and what are the pitfalls?
The fastest vaccine I think we ever made was the mumps vaccine, where that virus was isolated in 1963 and was a vaccine in 1967. That was amazingly fast.
4 years still, yeah.
4 years still. So that's it. The issue is it possible to develop a vaccine that quickly? And I think it would be fun actually to talk about what it takes to make a vaccine and what the pitfalls are.
Are you worried now that people are so scared of this coronavirus and what it's doing and our perception of it that they're going to rush into this and sign up for getting a vaccine that may have been rushed?
Well, yes, I think when people are terrified and they're terrified, we tend to have this break the glass mentality. And that break the glass mentality may mean that we're cutting certain corners, that we're not doing animal model testing, that we're not doing extensive so-called phase 1 or phase 2 testing where you do thousands of people looking for issues of safety and making sure that you get a good immune response before you even get to the efficacy trial. May mean that we're cutting certain corners, that we're not doing animal model tests, that we're not doing animal model tests, that we're not doing animal model testing, that we're not doing extensive so-called phase 1 or phase 2 testing, that we're not doing extensive so-called phase 1 or phase 2 testing where you do thousands of people looking for issues of safety and making sure that you get a good immune response before you even get to the efficacy trial. Yes, I worry that we might do this too quickly and miss things. But the other thing that I think is important here is that when you present a protein to the immune system, so we have two protein vaccines, the hepatitis B vaccine and the HPV vaccine, and they're great. They're safe. They induce an excellent immune response. The reason is that an HPV, human papillomavirus, is a good example. That's a virus-like particle. In other words, when the surface protein is made in these yeast cells, the protein in that vaccine looks exactly like it looks on the surface of that virus. That's critical.
Yeah.
The same thing's true for the hepatitis B vaccine. That too is a so-called virus-like particle. That protein looks in that vaccine exactly like it looks in nature. That's good. I worry, and I think we all are, I think everybody who's involved in this vaccine is concerned about the fact that that's not going to be this protein. This protein, whether it's an mRNA vaccine or a DNA vaccine or a purified protein vaccine or a vectored vaccine, may not look exactly like it looks on the surface of the particle. And here's why that's a problem. The goal of if this, see, there's my fist. If this were the protein, there's the business end of the protein. We'll say it's this. The part that binds to the virus. Okay, that's the business end. If you make antibodies to that business end, you will neutralize the virus. But you can also make antibodies to the not business end, to just the part that doesn't bind to the virus. Those are so-called binding antibodies. So you have the neutralizing antibodies and you have the binding antibodies. You want to make sure that the quantity of neutralizing antibodies that you have and the persistence of those antibodies is much greater than the binding antibodies, because the binding antibodies can be dangerous and cause something called antibody-dependent enhancement. And we've seen that. I mean, we saw that with the dengue vaccine. The dengue vaccine, as you may know, but the dengue vaccine in children who'd never been exposed to dengue before actually made them worse when they were then exposed to the natural virus, much worse. And causing something called dengue hemorrhagic shock syndrome. Children died because they were vaccinated children who were less than 9 years of age, who had never been exposed to dengue before, were more likely to die if they'd been vaccinated than if they hadn't been vaccinated.
Oh, wow. Yeah,
that's a problem, and that was because of antibody-dependent enhancement. Because what happens is those binding antibodies, they don't neutralize the virus; they just bind to it.
So the concern with coronavirus is if we make this protein incorrectly, it doesn't look quite like the wild type. We could be making the problem worse with the vaccine if we're not careful about that. Did I get that right?
Exactly. And so the trick is, the key word that you just said was careful. I mean, you need to make sure that you feel comfortable with this vaccine before you give it to tens of millions, and because this is a worldwide pandemic, hundreds of millions of people. So how do you do that? Typically with vaccines, what you do is you do an efficacy trial. So I think what has to happen is, if we're going to be doing this quickly, and we're doing it quickly, I think that I haven't seen animal model studies published on Moderna's vaccine. I haven't seen animal model studies published on Moderna's vaccine. I haven't seen that. All I know is that we did in Washington State a 45-person study that involved adults between 18 and 55 years of age who were divided into 3 groups of 15. And so I'm going to assume that I would have to believe that they would do thousands of people. Once they're clear that they have a dose, and they should do, I think, larger dose-ranging trials just to make sure that's the right dose, to make sure that they do a trial that represents the American public. So that you know that the trial that you've done represents the different percentage of African Americans, et cetera, in this. Then when you give it to the American public, you know that you've tested a reasonable percentage of the American public before you do that. If you're trying to get a vaccine out in 12 to 18 months, then you're going to have to skip some steps, major steps.
Okay. What are those steps that might be skipped?
So what would you skip? I think you skip the animal model studies.
It turns out that certain respiratory virus vaccines can trigger, through mechanisms that we don't entirely understand, something called immune enhancement. And in laboratory animals, it's been a problem for coronavirus vaccines.
I just worry that you would in any sense rush this, that you wouldn't do extensive animal model testing. If you're trying to get a vaccine out in 12 to 18 months, then you're going to have to skip some steps, major steps. So what would you skip? I think you skip the animal model studies.
Would we give it to children, or do we need to study them separately?
I certainly wouldn't give it to children until you are very, very confident that this vaccine is safe and effective, because children don't die from this virus. So you better make sure that you have a safe product before you put it into children. Right now, Moderna is leading the pack. This is the vaccine that Dr. Fauci talks about when he stands up in front of the reporters during these conferences. If we're going to be doing this quickly, and we're doing it quickly, I think that I haven't seen animal model studies published on Moderna's vaccine. I haven't seen that. All I know is that we did in Washington State a 45-person study that involved adults between 18 and 55 years of age who were divided into 3 groups of 15.